don't even know what that song is, but this is the weekly broadcast of This Week in Science. Welcome, everybody. Thank you for joining us. We're so glad you're here. Blair, how you doing?
Oh, I'm great. Let's talk about science.
Good. That's what we like to do. I hope everyone's ready. I want you to remember that this is the part of the show where I tell you if things go wrong, Blair has to leave. If things get edited out, then you see it all happen live here. The podcast is the edited version. So if you like things cleaner and tighter, make sure that you subscribe to us wherever podcasts are found. Go do that right now if you have not already, actually. And tell your friends to come and watch us right now because it's time to start. So share, like, let's get the ball rolling, shall we?
And if you like things dirty and loose, you just watch here.
That's what we like. Okay, it's all good.
Okay.
Goosey.
Sorry.
I see where you are tonight. Okay, starting the show. Woohoo! All right, starting the show in... Wait, I don't know what I'm doing anymore. Spreadsheet. There you are. I see what I'm doing. I have words. Dun, 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 dun. Starting the show in 3, 2. This is TWIS. This Week in Science, episode number 1016, recorded on Wednesday, May 21st, 2025. Do dolphins discuss science? Hey everyone, I'm Dr. Kiki, and today on the show, we will fill your head with space bugs, shrinking fish, and some teeth. But first, disclaimer. When lie after lie makes you question the sky and wonder what truth even is, remember to find the people who question themselves and don't try to sell you uncertainty or their life-changing supplements. Snake oil is greasy and easily slips past the lips of the charlatans and cheats. Remember to think about the source of that link before clicking your way past the brink. You hold the tools in your head to parse the good from the bad. And when you need more, remember This Week in Science, coming up next. And I'm on the right page right now, and I have to hit the button, and I'm scrolling, because usually I'm not the one. Here we go.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about science. And Justin is not here. Hopefully he'll be able to be here next week. But he's got some new responsibilities that are keeping him away from Wednesdays for the time being. Fingers crossed we will see him soon. Blair, we brought science, right?
Oh, yeah. I'm very excited.
Thank you all for joining us for this episode. We do have tons of science news. I did measure it out in the, using the metric measuring system. I have news stories about a new and improved CRISPR, some bugs in space, talking dolphins, and toothy origins. What's in the animal corner, Blair?
Oh my goodness. I have endangered species, I have clownfish, and I have bee brains.
At first I was like, this is a cool aquarium. And then I was like, one of these things is not like the other. Awesome.
I mean, you could submerge bee brains in water, I suppose.
Fish food. That would feed the fish. Okay, moving on forward. Everyone, I hope you're excited about this show tonight. As we jump into the show, I want to remind you that subscribing to the TWIS podcast on your favorite podcast platform, YouTube, Facebook or Twitch is the best way to keep up with this show every week. And whenever we publish it, because sometimes we do it more often, sometimes less often. It's a very interesting thing. We try to be every week at least, though. We're here Wednesdays, 8 p.m.-ish Pacific time, to do a tight 90s show, and you can subscribe and be alerted every time a new episode is published. It's really good. You could get your friends to subscribe, and then they could be alerted every time a new episode is published. Whatever. If you need more information, go to TWIS.org. You can find show notes, link to our Patreon community, also to our Zazzle store, TWIS.org. But now it's time for the science. You ready, Blair?
So ready.
So, you were born ready. I know. I see you there. So I'm going to start off the show with just what everybody wanted. New and improved CRISPR.
Yay.
Woo-hoo. So we remember CRISPR, right?
Of course.
Right? Yes.
Gene
Of course
editing.
we do. Yeah, but CRISPR, it has caused a big, not tumult, hubbub, I don't know, lots of movement in the
Controversy.
that too, exactly, in the gene editing world. CRISPR, sometimes called molecular scissors. CRISPR came from bacteria. Researchers, long time ago, they were like, oh, look at these cool tools the bacteria have. They go snip, snip, and then they're able to add new, what is going on here? And it, on and on and on. And eventually, researchers were able to use this CRISPR tool along with another enzyme called Cas9, and they've come up with other enzymes since then to create more and more accurate methods with which to edit genes. The hope is that we will be able to edit our own genes to be able to get rid of disease and all sorts of issues are that CRISPR-Cas9 tends to not be as accurate as we want. And so in the scissor cutting the DNA part of the whole issue, the whole process, sometimes they don't cut the right place. And so you have what are called off-target edits. Sometimes once it's cut and a gene is put in a place, sometimes there are errors in the repair process because the body, the cells have its own repair process that this CRISPR-Cas9 takes advantage of, so that after a gene is newly edited into, or even single nucleotide changes, after they're edited, they might not be repaired correctly, and sometimes that messes up how the resulting proteins or the resulting processes work. So one of the researchers who was in a big, who was also involved in pushing CRISPR-Cas9 forward at Harvard University, his lab was also involved in a big patent fight against the Berkeley lab with Jennifer Doudna and Emmanuelle Charpentier. And so this David Liu, his lab at Harvard has just come up with a new, not Cas9, but a new little sidecar for CRISPR, and it's called Cas. And it too.
Can we pause? Sorry, you sound like you're underwater.
All of a sudden?
Yes, and now you sound fine.
Always? La 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 la. La la
Now
la.
you sound great. Okay, good. Continue. For like
I'm
3
talking with
sentences,
dolphins.
you sounded like you were underwater.
Weird.
So I was worried something was wrong. Sorry, continue.
Thank you, Blair.
So there's a new sidecar called Cas.
There's a new side. Yeah, no, Cas is the old Cas9, Cas5, Cas11. Like that's the old sidecar. Now they're using it for other things, but there's a new sidecar that's been developed, just published in Science this last week, with an associated enzyme called a CRISPR-associated transposase, Cas. So these Cas.
That so I didn't hear you. So I was like cast, but that's what you said before. Cast.
C-A-S-T.
Wow.
Cast.
Okay. The branding.
Sorry.
I take issue with the branding.
Yeah,
It's too similar.
Cast. I must I'm supposed to, in radio, you're supposed to mind your P's, Q's, and T's
Yeah, you don't want to pop.
for the popping, the plosives. Anyway, this CRISPR-associated transposase also comes from bacteria, and it is used within bacteria for jumping genes. So these CRISPR-associated transposase, they're already in bacteria with CRISPRs. They work with, as an enzyme, with CRISPRs to help these jumping genes move around within a cell, right? I don't want to be here anymore. Let's jump on out and across the chromosome and go jump into another part of the genome. And this CAST system is what helps it do that. And so they were like, that's great. What they discovered, though, is that human cells don't like CAST. And so they used a focused evolution process where they pushed the CAST enzyme to exist and survive and be able to work with CRISPR within human cells. And so this approach they used is called PACE, and it's
more acronyms.
It is. I'm not even going to go there. But basically, each round of evolution, each generation, there's little tiny, tweaks, mutations, etc. And eventually, what they've done, they say over 200 hours with PACE, which is basically several hundred evolutionary generations, say the researchers. And if they were doing conventional methods and not using PACE, it would have taken years and years and years, but this took just over 200 hours. It's fine. So they have this evolved cast. They're now calling it EvoCast, and it has 10 mutations that have come up. It can work well in human cells, and they were like, let's try it out. And so they tried to see how EvoCast and CRISPR would work to basically do some gene editing in cells, and they found that it increased. So previously, when they tried to use CAST in cells, it was like 1% that actually worked, and this time it was like 12 to 15%. So they've 100x'd, 200. They've really amped up how well it works. It still doesn't work really well, but they see
still doesn't work. It's better. Still doesn't work. Closer to working.
We're gonna edit. Yeah, we're gonna edit your genes, and it may or may not work. So, it's still not ready for prime time, is where they're at. But what the methodology they're using has done is created a potential new, not as scissor-ish way for the gene editing to take place. So because of the way CAST works and the way it integrates genes or whatever they're trying to put in with the CRISPR system into the genome, you don't have to rely on the same kind of cutting and then repair. And so the accuracy is better. There are fewer mutations or errors that occur in the repair process. So they have lower accuracy in terms of actually getting genes in, or efficiency, I guess, but it's better. So hopefully they'll be able to improve how well it works, to be able to get it to the right cells in the body. Maybe they'll be using it to edit like immune cells for CAR T cancer therapy. So researchers are pushing forward, improving our ability to edit everything.
Nice. That's interesting.
Yeah, you think it's nice?
CRISPR's been on the field, I guess, for a pretty long time. And I think a lot of promises were made very early on.
How often does that happen, Blair?
Of course. But I think that it happens a lot. But I think the difference is that in this case, they're like, oops, we overpromised and underdelivered. And so this other kind of company, group of researchers, whatever you want to call it, it's like, what can we take from this very lofty promise? What can we tweak? And what can we adapt to actually try to get somewhere? It's surprising to me it's taken this long. that for somebody else to look at CRISPR-Cas9 and go, okay, what could I tweak here? Especially with, what could I tweak here to have my own proprietary product? That's the other piece that I'm surprised has taken this long.
That's the thing. The way that the patent fight came out, this lab actually won most of the patents for the CRISPR research, even though Doudna and Charpentier were the first to publish. Anyway, there's a whole thing. Controversy.
There's lots of like sci-fi dystopian things that we could think about with CRISPR. Of course, you can always go there, but I like to assume that because if I can still be jaded, there's so much money to make in
Blair,
medicinal approach. Oh, absolutely not. But there's optimism in my jaded state, right? Where, there's a lot of money to be made in the medical approach of using gene editing. So
sure,
I'm optimistic that they will find a way to make this work to help keep people healthy longer because there is money to make.
I mean, right now with the system we have, it's a good reward, right? Like, help
absolutely
people make money is good. Do good, have money. That's fine. Anyway, I don't think they're getting all the money yet.
No, but they could, which is why they're scrambling for the patent.
So, on the line of CRISPR and this new cast sidecar, there's also another story this week that I thought was interesting, and you're going to love it because it's the world's first gene-edited spider.
Ooh, okay. To make it more friendly?
No.
Oh. To make it extra fuzzy?
No.
Oh, okay. What for? 6 more guesses? I could do 6 more.
No. This isn't
extra legs. Less legs. Oh, okay.
No. What thing about spiders are we like, hey, this is cool and we could maybe use this for like people technology sometimes, like.
Oh yes, silk, of course.
Spider silk. So what did they do to the spider silk? They incorporated a gene into the young developing actually not developing. I think it was I think it was before there were actually eggs. I'm not sure. The eggs were there, but before anything happened to them. The
unfertilized eggs. So you're saying?
Unfertilized. Thank you. I forgot words. So the offspring got a gene to spin silk with red fluorescent protein.
Red fluorescent.
So the silk, yes. So the silk that was made by these spiders is red fluorescent silk.
Why?
Well, number one, spiders have been neglected in terms of gene editing.
Okay.
We talk about Spider-Man. What about Spider? So
there's Spiders Man. Did you know that? That is a man made of many, many spiders. There's also Man Spider, which is a man-sized spider. So there's lots of variations in the Spider-Man universe.
I know.
And actually, Spider-Man turned into Man Spider because of gene editing, I will tell you. I do believe that is the story. The chat room can correct me if I'm wrong, but at least in the cartoon.
How often has it gone the other way where the spider was gene edited and then turned into Man Spider or Spider-Man or Spider or whatever, right?
Right.
Spiders have been neglected. And so these researchers, felt sad and said, let's do something cool. And so they gene edited the spiders. But also because spider silk is one of the interesting parts of spiders that we pay attention to, and that if we can gene edit the silk without messing up its its formation or construction while it's being spun, then that's actually kind of a neat thing. Like, what could we put, what other proteins can we put in that don't destroy the tensile strength or the potential uses for spider silk? So they used CRISPR-Cas9 to do this.
Interesting. So in what scenario would we want to edit the nature of spider silk?
When you want it to warn
Be
you
red
ahead
and
of time before you
fluorescent.
walk in.
Yeah, so if you make it so you can see it, then it defeats the purpose of them actually being able to catch things.
Yeah.
So that's not
the
so
other
good.
Yeah, the other side of this story is they were not just interested in the silk fiber engineering, they were looking at other ways that they could not just knock in a gene, but also knock out a gene. And so with the CRISPR-Cas9, they did a knockout of the gene Sine oculus, which resulted in spiders without any eyes. So
What? No,
yeah, I don't like that part of the study.
that's not nice.
Yeah.
I mean, I
Nice,
guess,
Val.
okay, so in thinking about all of the editing we do to mice, right? And all of the knockouts that we do in mice to test
So
conditions.
many, and how often do you go?
I could see, okay, we knocked out the eyes in some spiders to figure out how much they use vibration in communication and navigation or something like that, right? Like you could, if I forget about just the optics, of this, sorry, and I look at it in terms of how we use other scientific models, like fruit flies, like mice, right? We do this kind of stuff all the time.
But
Just saying that you had them grow up without eyes seems so wild, but yes, there is some scientific precedence for doing things like this. So
I mean, part of it is, number one, like I said, the spiders have been neglected. So it's proof of concept. Does this work? And how does it impact the developing organism? And what they were able to show is that they understand enough to successfully be able to have spiders develop and live lives with spinning red fluorescent spider silk or not having eyes. And as much as it's like right now, you go, what's going on? The next question is, if we understand how these things work in spiders, what more can that teach us if we start looking at it very specifically in terms of knocking genes in or out to study evolution, development, like to ask questions related to comparative biology. How do these organisms, which are the same genes that make these particular traits happen in spiders exactly the same as crabs or humans, or how does it all work? So there are specific questions that can be asked if this process works properly. And now we know it does. And we don't have to neglect spiders anymore.
well, now we have red fluorescent silk. And I will say, Fada in the chat room asked, haven't we brought stories before about spider silk being used for all sorts of new purposes? So occasionally, but the grand majority of stories that I have seen related to spider silk is about studying silk to be able to make similar substances artificially. Not about like farming spiders for silk for human use. I feel like that is not generally the narrative. It's like we studied the silk to figure out about the tensile strength and try to replicate it.
And like as it's being formed in the spider, there are very particular proteins that come together, and there's a very specific chemical process that leads to the silk that comes out. And so for ages, it's been, can we create spider silk? And we couldn't for a really long time. And I think it was like a few years ago when somebody actually was like, oh, I think I might have figured it out. I don't know. Nobody's wearing spider silk shirts right now that I'm aware of.
No, no.
But maybe they would if they were fluorescent red. Yeah. No. But anyway, yeah, CRISPR-Cas9. So fascinating. What do you want to talk about, Blair?
Oh, I have bad news, unfortunately. It's
At least
and I
you
have to
told
talk.
us right up front. I got
Yeah,
it.
I'm not gonna mince words here. The Trump administration is gonna kill a bunch of endangered species. That's ultimately what's happening here because they are gutting the Endangered Species Act.
Mm-hmm.
And I wanted to bring it up because this is where conservation science and politics do collide. This happens. And we do need to talk about it because there is going to be some fallout. So the Endangered Species Act passed in 1973, and it bans some of the kind of legalese is the take of endangered species of fish or wildlife, including harming protected species. And in 1975, they had to define harm legally. What is harm? So take is very clear. Did you remove that animal, alive or dead, from their space? But what is harm of a protected species? And so they included the number one cause for animals to be threatened or endangered, which is
People
yes, but what activity that people do?
building houses, living.
Yes. So usually, like when I used to teach our biodiversity basics programs in the Zoomobile, the kids would always say, hunting. Hunting is the number one cause. Nope. Like pollution. Nope, it's not pollution. It's habitat destruction. Habitat destruction is responsible for 81% of animals being listed as threatened or endangered. Direct hunting or poaching is only about 17%. Sometimes it's less, depending on how you count things. And so habitat destruction is the number one cause of threats to protected species. And so since 1975, it has been clear that habitat destruction is included in the Endangered Species Act. Now, in 1995, there was a Supreme Court case, Babbitt versus Sweet Home Chapter of Communities for Great Oregon, which the Supreme Court ruled that the definition was reasonable and allowed federal agencies to continue using it. But now, the Trump administration is planning on changing the definition of the word harm to leave out habitat modification. And so the plan is essentially to create free rein to develop on protected animals' habitats. There's about 107 million acres of land in the U.S. designated as critical habitat for endangered species. So they're not listed as protected open space. They're not listed as a national park. They are listed as designated critical habitat for endangered species. The only reason that 107 million acres of land is protected from development is because there is a protected species in that space. And so the second you change this definition of harm, that is when these lands are open for drilling, development, any number of other things, logging that different industries and stakeholders would have interest in using. And so this isn't in effect yet. It was open for public comment. The kind of the glimmer of hope I will give you is that in the about month and a half that it was open for public comment, it closed 2 days ago, 345,000 people left comments urging the government to not do this. Now, I don't think that's necessarily going to do anything, but that means that the general public is very much in support of the Endangered Species Act. There is bipartisan support for the Endangered Species Act. People generally all want to keep it and think that it is a good thing. And so the general public have really spoken out about this and demanded that this not occur. Unlikely that will cause anything. Likely this change will still go into effect. The other interesting piece is because of that 1995 Supreme Court ruling, there will be legal challenges if this does get changed. But the problem is in the amount of time that it takes for that legal challenge to be seen and heard. And even if this change is overruled and returned to the original definition of harm, there is no guarantee that land will not get sold and developed in that period of time in between.
Right.
So I tell you this, I know it's too late to do anything about it. People have already left their public comments, but just so you are aware what is going on and how there are people working behind the scenes who actually are pretty smart and know how to tweak specific language legally to cause real harm to the positive progress we've made towards the environment. Harm, real harm. Yes, the actual meaning of the word harm, right? So I just bring it up so that we know what we're dealing with. So we recognize that this is an area of concern. If you have the ear of your local politician, go ahead and call them and shout about this. Congress wrote the Endangered Species Act. They wrote it. And I have where is it?
With a lot of help from scientists.
Absolutely, yes.
And
so it went, go ahead.
Bipartisan. It was also bipartisan.
Yes.
This is not just, one side or the other. This is Congress.
Yes,
together.
and so in 1973, Congress wrote, when they enacted the Endangered Species Act, that it is to, quote, provide a means whereby the ecosystems upon which endangered species and threatened species depend may be conserved, and to provide a program for the conservation of such endangered species and threatened species. So Congress wanted it, the general public wants it, the Supreme Court ruled in favor of it, but there are the powers that be that want to just unilaterally do the right thing fiscally for themselves. And so I had to daylight it. I don't really have a very high bright side right now, other than to hope that it either somebody steps up and recognizes how unpopular this is and it does not go through, or that the legal action resulting goes the right way and proceeds quickly so we don't lose too much land. Those are the two things that I can hope for at this point, but this is pretty bleak.
And I want to come back and say this is the downside, right? This is the bleakness. This is are we going to be able to do the right thing? Can we put up pressure against these movements? This is not a said and done thing. Aside from the comments, this is the part of the civic act, part of science, right? Where you're involved in the process of how science is integrated into public policy, how everything moves forward. It's talking to your representatives,
Absolutely.org.
There are lists of things that we could be calling about every single day. But this is one of those things where I think it's important too. Law, like science, has its own definitions of words. And it's only through those definitions that we have this very specific way of focusing on information and being able to speak to each other in a very accurate way. But when you use that, not as a tool to make things better, but as a weapon, then it's something that we all need to be aware of. And it's just a theory, right? We've been talking about words in science for so long, and this is where that is important, where people need to know how words matter. Not just the way you understand them, but the way that other people use them. So I think even if change is made, it was one way once, it can be one way and it can be another way again. So even if something happens to change this, and it's a slow process, there's a whole bunch of stuff that's going to need to be fixed. But this is one of those things that I don't think people will back down from. So
No, the public will not allow this to go quietly. I mean, all of us will not allow this to go quietly. The Endangered Species Act, this is more than 50 years of conservation work that has completely turned the tide on species in this country.
And so, but I think the thing for anyone in the audience who wants to have a conversation with people about why this matters, if we continue to destroy diversity in ecosystems around our planet is going to, it's already moving certain ways. Species are dying, Anthropocene, yada, yada, yada. But if we prioritize our uses, our wants to take things, I'm going to use the word take things out of the land for our own uses over being able to exist on this planet in the long term, what we're doing is we are going to shorten the ability of humans to live on this planet. Might be great short term, but it's certainly not going to go long term. So preserving diversity preserves ecosystems, preserves biosphere, preserves life on Earth generally. And yeah. Am I wrong, Blair?
No, that's 100%.
I mean, it's like people are like, I need that butterfly to move so I can build my condos. We need housing. Housing's great. But we have to figure out how to make things work in a more integrated way. How can you do development in a way that maintains ecosystems, habitats, and doesn't destroy? How can we meet our 30% goal, right? How can we...
We've found a lot of ways. I think that's why I'm feeling so frustrated by all of this, is we've made great headway in the last 50 years. We've brought animals back from the brink of extinction. We outlawed DDT. We brought back the California condor. We brought back the bald eagle. We brought back the American alligator. We did all of these things through the Endangered Species Act. And
Blair,
we built tunnels under roads.
But you stopped people from mining and using land the way they wanted to. So people couldn't do stuff with the land that would make them money. So,
yeah.
I mean, come on.
Okay, but what about this? What about all of the people who work in environmental compliance? What about all of the people who monitor endangered species around construction sites? Those are all going to be people that have less jobs now. So that also impacts the economy. No, because it is required because of the Endangered Species Act. So you still have to do environmental assessments, right? So the second you don't have to do that, there is an economic impact to that as well. I think that's the piece that always gets left out of this conversation is conservation does provide for the economy 100%. There is a huge amount of study around how biodiversity creates wealth for a community. And when you remove biodiversity, wealth actually decreases in that space. And this is where you're not measuring the true cost of things. We're not measuring the impact of these large boom and bust developments. And we're not measuring the actual financial gain of conservation. And so it's put as a cost-benefit analysis, right?
It's not the people there. It's not the priority they're caring about. So it's it's
lobbying. Basically,
it's lobbying, and it's the way that people are misusing science. It's cherry picking their information. They're not taking the full story.
Yeah, absolutely. So they're putting their thumb on the scale, right? Because they're saying, oh, well, if we build this development, that's feeding the economy. But they're not looking at the fact that they're losing biodiversity, which costs money, and they're also eliminating conservation work. So it's totally unfair to look at it as a zero sum.
And why that location? Why aren't there other?
Maybe because there's already plenty of housing in the United States for everyone to own a house, but it's all been bought up by corporations. So you actually don't need to build any more houses. What about that?
I don't think that's actually true.
Yeah. Yeah.
I mean, there's a lot. Yeah, there's a lot held. I know that. But yes, that's a whole other game. We're not going to get into that economics. That is a debate, and I would need my list of facts next to me to have it. Oof. Oof. It's getting hot in here. Housing is the big thing right now. Like, yeah. But by moving backwards, by redefining the word harm for the Endangered Species Act, we would be moving our society backwards. That word being changed in that instance is also one part of a landslide that changes the way that they're able to get all sorts of things changed. I don't want to go backwards.
No.
I don't want to go backwards.
But we're doing a lot of that lately.
I know, but I think that sometimes, go forward and back, forward and back. That's the whole 2 steps forward, 1 step back, 2 steps forward, 1 step back. But we have to make sure we keep working toward that step forward, that next step forward, and not get caught in the feeling of going backwards as if that's where we're going to be forever. Because this is a moment. That's all. It's a moment. And I mean, we're not even talking about the big, beautiful bill and the addition, the poison pill within the big, beautiful bill to make sure that AI can never be regulated. Have you heard about that one?
No.
Yeah. So the big, beautiful bill that they're going all night for recently has several poison pills, one to cut Medicaid, another that basically says AI, no state can regulate any AI technology. Ever.
I don't like that.
And there are little things, it's a Trojan horse bill. There are things in there,
God.
and take a look, everybody.
And that's the plan.
Because it's a Trojan horse bill. I mean, that happens with every bill. Stuff gets shoved in, and then you're like, what? How is that happening? But if they get it in, and then the methodology to fight it later is not there because they've changed rules and various things, it's an issue. So everybody, pay attention. Do you want AI to rule your life? Do you want harm to not really mean harm? But what about bugs in space?
They need really tiny helmets.
That's really funny. Now I want to see that as an animation or stop motion. Might be kind of funny. Researchers have been looking at swabs from China's Chang'an space station to look for bacteria. Like we've been talking for years. This is usually a story that Justin would talk about. Talk about how there's so many bacteria in clean rooms. How we have found bacteria on the International Space Station previously. Well, now China's space station is part of the club of having a new species of bacteria they've discovered called Nealia tiangongzensis. And this species is related to an earthbound strain known as Niala circulans, and it's usually a rod-shaped soil-dwelling bacterium, and it's kind of thought of as pathogenic, a pathogenic form of bacillus. Anyway, these particular organisms and the others like that were found on the ISS, the bacteria that evolved, they are extremophiles. And one of the things that they do is they form spores so that they can exist and survive in the difficult radiation, gravity, like all the different strains, not strain of bacteria, but the stresses of space. And so this particular Nealia tiangongzensis it's been published this last week, its genes and functions, and it's lost basically all sorts of genes that its relatives normally have and others that have to to digest stuff and get energy. The one thing it has is a unique mutation that allows it to break down gelatin and use gelatin as a source of nitrogen and carbon. And that's really great because then it can use that to create like a biofilm, which makes the bacteria stronger and more able to survive in space. The
That's
one
their
thing
helmet.
they don't know is their helmet, exactly. They mutated to make their own helmet. One of the things they don't know with this yet, because Nealia is related to Bacillus, and Bacillus can cause sepsis, and its earthbound relative can be kind of, can be toxic. They don't know how bad it is or good it is for people in space. So this is one of the interesting questions and why they're doing these studies, because you think you're sending a clean ship to space, but basically you're sending a ship to space with bacteria that have a very survivalist background. And so the question is, in addition to their new mutations for just existing, if they are related to or come from pathogenic backgrounds, what does that mean for these new evolved strains that exist in space? And what does that mean about the panspermia that are stations, our space stations, our spaceships, everything? What does that mean about this panspermia of Earth organisms into space and how they exist with humans?
It just means it can't be avoided, which I think we already knew, right?
But are they extra pathogenic? Like, do any of the mutations?
Right.
I mean, so far,
Right.
nobody's been like, I breathed and I have sepsis. Like, there's nothing like that.
Right.
But,
Yes. Yeah, I feel like if
and,
that would have been a problem, it would have happened already, right? Like, we have people living on the space station. We've sent people out to space enough that I feel like people would keep coming back with super bugs and be like, what the heck happened to you?
I
Getting
went to
sick in
space.
space.
I couldn't work out in it. I got immunocompromised because I was in space and couldn't work out enough.
Yeah, like, do you know what I mean?
Cold.
It's the sample size isn't enormous, but it's also not tiny of the number of people who've been to space.
I mean, any one sample set is pretty small, but over
Right.
time.
But over time,
is
I'm
also-
saying the number of humans we've shot into space is
But think about it. It's like they're evolving and we just keep sending different people. And
right.
It's like,
Yeah.
I don't know. I think it's very interesting.
I don't know. I don't know. I mean, what's the solution? Because our clean rooms can't handle it.
Well, that's the question, right? How do you actually create a really clean room? Because
An
these
atomic
extremophiles,
bomb.
and you're no, not going to space, guys.
A teeny tiny one. in the room. It's insulated in the room, right? I don't know. Yeah, that's the problem, right? Like,
Really
What-
strong scrubbing bubbles. I don't know.
so I guess this is identifying a need that now more research has to go in how to make a better clean room. That's really
Or-
what this story is about, right?
It is, but it's also it's either or. How can you make a better clean room so that we don't take more extremophiles to space? Or how do we make sure we coexist with these new species in a way that doesn't make humans sick? And can we? Can we do it? And I think if we can't, we shouldn't be taking them with us to other planets.
No.
That seems like
Just take their helmet away. Kiki. Hold on.
Were you drawing little space bugs? I know.
Yeah, here, let me turn off my blur.
It doesn't want to. It's going to blur everything because even though it's in the foreground, it's in the background.
There you go. Ooh.
Ah, it's a cutie. Aww. I think we should make a sticker. That's really cute. Space bugs with helmets. I love that. That's really cute. I'm glad that I turned into like a college lecture for you.
Yeah.
Oh my goodness,
Started
you're like,
doodling in the margins. Yeah,
I gotta
absolutely.
do. I gotta...
I don't know. I started thinking about germs wearing space helmets. It's funny.
It is funny, and I'm really glad you went with it. And you had a pet paper and a pen next to you. That's amazing. All right, everybody, we're here, and so are you. Thank you for being here for This Week in Science. Tell your friends about the fun times they can have with Blair drawing little pictures of space bugs and helmets. If they were only here too. Oh my goodness. Please tell your friends to subscribe. The more subscribers, the more we can talk about using science, how science is a part of our lives, how science is around us every day. And we have more and more people to talk about information and how we use it, how we analyze it, how we come across it in our lives, and who we trust. So I'm really glad that you trust us. I'm really, really glad. Thank you for being here. And, oh, by the way, if you would like to support us, we are listener supported. So head over to TWIS.org, click on our Patreon link or the Zazzle link, and help us out. Thank you for your support. We can't do it without you. All right, Blair. Are you going to draw through the Blanimal corner?
No, I can't possibly do that. I was just adding some. Is it going to show you some planets and stars? No. And I wrote space germ.
I love it. I love that so much. All righty. Well, you all know, coming back with more TWIS right now for the second half, and it's time for Blair's Animal Corner with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels, there's Animal Corner.
What you got, Blair?
I have shrinking clownfish.
What? Are they balloons?
Yes. So this headline caught my eye because normally when we talk about animals shrinking, it's figurative. And it means that over time, they're not growing as large. So generationally, the species...
Like the island effect.
Is shrinking. Exactly.
Yeah.
Absolutely. Okay. But in this case, individual clownfish are shrinking. They are growing up large, and then they are ending up smaller, and then they are growing large again. So they are like losing body mass and size.
Why? What?
Yes. So this is from universities of Newcastle, Leeds, and Boston. And they were looking at clownfish. They had 134 fish. 100 of those shrank when exposed to heat stress. They looked at them over a period of 5 months. And so they found that this phenomenon was actually very common. The only other example that I was able to see cited in the study was actually with marine iguanas, where it sounds like during similar environmental stresses, the marine iguanas will actually reabsorb some of their bone structure, some of their bone matter, and they will also shrink. So this actually has been recorded in the animal kingdom before. I wasn't even aware of that. But this is crazy because clownfish are already crazy because they're sequential hermaphrodites. They start out male.
Okay, those words. I know. Oh, right. They're crazy because.
Yeah, they're sequential hermaphrodites. So they start out male, they're tiny. And then there's however many females, right? They're large. And then if a female dies or leaves and there is space for a female in the area, then the male will get larger and larger and larger, develop female body parts, and transition to become a female individual, and then lay eggs. So the age-old kind of joke from people who know about clownfish and their sequential hermaphrodism is that Marlin in Finding Nemo should actually turn female throughout the course of the film because the mom dies at the beginning. So should actually progress and change. So the reason I bring that up, it has nothing to do with this study, but it's relevant because there is proof of the plasticity of the body plan in clownfish. They have other mechanisms to completely change the shape and function of their body throughout their lifetime. So I would say out of all the places to look for this weird adaptation where you shrink because of environmental pressure, it makes sense it would come from a clownfish. So I had to dig through the
plasticity, but shrinking is different from.
Yes. So okay. Yes. So I looked through a couple different press releases and I dug through the actual paper and I was trying to figure out the mechanism and why heat stress would cause you to shrink, which generally speaking, climate change is causing animals to shrink. But again, on the scale of generations. They're not growing as large just because they have environmental pressure, and so there's more demands on their metabolism. That could be that they can't find as much food because the climate is impacting plants, or it could be because the heat is causing them to sweat and lose energy through sweat, right? If they're an animal that could sweat.
So physiologically, like body surface area, like is a big deal when it comes to heat and heat transfer and heat loss. So that would be one big thing. But this is fish. So I'm like, there's this, they live.
Right. So one thing that they called out in the paper that I found very interesting is called gill oxygen limitation theory. And so cold water carries oxygion. Hot water carries less oxygen. And so if the water is hot, there's less oxygen. But this is where this gets really interesting. Gills are essentially two-dimensional. They're flat. And when you increase the size of a two-dimensional object, the surface area does not increase at the same rate as a three-dimensional object. And so the three-dimensional body size
increases
the
more
overall than the gills. So the amount of body that needs to be oxygenated outpaces the size of the gills increasing. It's very interesting. And so that is one of the main hypotheses of this research group, is that is kind of the main stressor that they're feeling. Of course, there's other things. They could have changes in food availability, as I mentioned. The content of the food could change, and the effort to find food could change. There's lots of things that could be impacted by climate, but I think this gill oxygen limitation theory is a really good one to kind of explain why this might be happening. Now, how they're losing body mass, they do not know. This is totally weird. They think it might be a hormonal trigger, but that still doesn't explain the mechanism by which the body shrinks. Is it eating itself? What part of the body is getting reabsorbed? There's lots and lots of questions to be had here. So this happens when there's like extreme heating in the water. And so they measured the lengths of these fish for about 5 months. And during that 5-month period, they shrank. So.
Yeah, but then, so there's that, but then there's the, like you said, where did their food go? Was there enough food? Were they just not hungry? Like they just wanted to drink cucumber spritzers?
Right.
Like,
so that
would like it's hot.
That would prevent them from growing any larger. But see, that's the thing. Their length reduced. It wasn't just they're lighter. They didn't get skinny.
No,
they shrank. It's as if you lost an inch. And it's not, you didn't change your posture, you actually lost an inch. What is the mechanism there by which you lose height?
Oh, I know. They're like shrinky dinks.
Yeah, because it's hot. You're right. Yeah, it just got hot. They shrank in the dryer, basically. Yeah, so I think I will be interested to see if this research group continues to study and bring some things into the lab to try to replicate this, or I don't know.
Well, see, now I'm curious from just the morphology perspective of like, hey, length changed, but what about like their spinal column? What about like what was the distance between vertebrae? Like what happened?
Yeah, did they just lose cartilage? Right. Great. Yeah.
Is there a limitation to what can be lost for them to shrink? Like, or are they like the incredible shrinking fish and they just are like, I get smaller the hotter it gets?
Well, and the question that I have from a conservation standpoint is, how long did it take them to get back to the size they started at? Like, did they rebound once temperatures regulated? And what was the long-term impact on the population? Because, again, the fish that can grow up bigger become female. And so if you have a size impact over time, does that impact your sex ratios in your fish?
How does it change mating? Reproduction.
Does it change how many eggs they lay because they weren't female as long? Like, there's so many ways you could extrapolate this.
And they're interesting also, like you said, because of the relationship between the male and the female and the guarding the eggs and the way that they can switch. How does it change the behavioral dynamics of the animals? So if the population's changing, you have fewer females, more males because of limitations in sizes, not just number of eggs, but then fewer females. Is there more competition between males? And then with that, like, yeah, exactly. How does it change everything? Does it change the way that females accept males? Like, I don't know.
Yeah.
This is fascinating, Blair.
It's really interesting. They did a little bit of social research with these groups, and they did find that the mating pairs didn't get disrupted. So it seems like they stayed enough of the same size that it didn't impact what sex they were or their sex organs or anything like that. But it was also short. It was only 5 months.
But that in itself is interesting. So they're like maybe shrinking together. Like, was there correlation between the mated pairs? Did they shrink together? And like you said, there's hormone changes that lead to the sex differentiation that lead to these things. And
yes. So one of my favorite things is they have a better chance of surviving. Yeah, go ahead.
I was just gonna say, if the heat stress doesn't impact, it's not changing the hormone balance that would normally impact, or maybe it's even blocking the pathway that would impact the sex transition. Like, I don't know. I wonder how that all works together.
You're onto something, because they did a lot of interesting social interaction studies alongside this too, and they found that the survival rates were the highest when they shrank alongside their breeding partner. So they both shrank. So they kept
that shrinks together.
Yeah.
Stays together.
So the other piece of this that I think this reminds all of us to look out for in the future is that what other animals are shrinking? What other animals are changing their body plan in some way to respond to environmental impacts in a way that we haven't previously assumed they could? Like, it felt like a one-way street. You grow up, you don't grow down.
So what I wonder is, like, clownfish, you said they have this variable body form already, but like, what if what we're seeing, because we didn't see so much of the sex transition at the same time, what if it is other fish species like changing sizes? And how common is that across species?
Yeah.
Clownfish, though, are really interesting because they're coral reef species.
Yes.
So they're important to the whole coral reef ecosystem, which is impacted by high temperatures. So that's the next step of it.
Yeah, they keep those anemones healthy. Yeah. Let's end on some heartwarming bee brains, shall we?
Heartwarming bee brains. Okay, go for it.
Yes. I thought you'd
Too many
really enjoy
organ
this,
systems.
Kiki.
Too many organ systems. Hearts and brains. I don't know.
Bee brains, which are, you know, they're about the size of a grain of rice. They're pretty complex, but they're still also not a lot of neurons. It's a very small group of neurons.
It's actually not a brain, it's a ganglion.
Yeah, it's like a nerve ball, but also it's kind of a brain because it still has certain structures. It has optic lobes, it has a central body. So like there's brainy things about it. So for ease of discussion, I will call it a brain for this story.
I will accept that.
Great. A study from the University of Trento suggests that bees have sleep similar to humans, and that in fact we could use bee brains to learn about human sleep. We know bees sleep. We know bees will go into torpor when it's really cold. But the mechanism and then the individual brain activity that happens during bee sleep has not been well studied. And what researchers found is that sleep appears to carry a similar signature at a neuronal level to humans. Not too surprising. I feel like sleep is pretty...
It's common. So some
consistent.
aspect of rest cycles are common in every living creature down, like from vertebrates to yeast. Like there is an active cycle and an inactive cycle that can be identified as something quote unquote sleep. Yeah.
Right. So the researchers combined optical brain imaging. They used imaging on these little bee balls and nerves, machine learning analysis, and computational neural modeling to study how sleep influences the neural networks responsible for sensory perception, specifically in the antennal lobes, which are the olfactory centers, smell centers. And so they had data collected during the night when bees naturally sleep. Their body movements were monitored with a front-facing camera. Their brain activity was simultaneously recorded with a 2-photon microscope. They analyzed the calcium concentration in neurons, and by doing that, they were able to detect neuronal activity during sleep and wakefulness. Don't think too hard about the mechanism of this research. I don't want to talk about it. I kind of started thinking about it and went, no. So they showed that in resting states, you know what I'm talking about, Kiki. In resting states, the brain networks of bees, they switch to synchronized and reduced information processing, which is like what happens in us. So, for example, if you look at a single parameter like smell, the synaptic coupling between neurons caused the brain to no longer decode odor signals. So they reduced olfactory perception just like it does for us. So they saw that basically they went kind of numb to stimuli around them that they would normally notice during sleep.
Like our deep sleep cycles, right?
Right. That you can sleep through crazy things happening around you. And it's because a lot of things are shut off, or you'd wake up at every gosh darn thing. This is cool to learn for bee-specific reasons, of course. They get to look at pollinator health. They get to look at ecosystem stability. They look at potential impacts on bee sleep and how that might impact bees' effectiveness to pollinate, yada, yada, yada. But the other thing that it seems like researchers are really stoked on related to this is that this creates an opportunity to study sleep at the level of individual neurons in a sample species that has a much simpler brain than humans. And because they're seeing a parallel between what happens in our brain and what happens in a bee brain during sleep, they can study the individual neurons in the bee brain to learn more about sleep disorders in humans.
Yeah, I get that. Like basically, the bee brain in this scenario becomes a model system that can be tweaked.
Mm-hmm. Absolutely.
Turn stuff up,
Yeah.
turn stuff down. Yeah.
Yep.
What makes a bee an insomniac? What makes them wake up multiple times in the night? Like, yeah.
Yeah. What makes every little sound or smell wake up a bee when their friend bees don't do that? Or what makes this bee impossible to wake up?
Like my child on a school day.
Goodness. Yeah. Yeah.
No, I think this is this is one of those studies where you go, this is like with the spiders, like what, why, and what they are discovering is a simplistic model where you've got 100,000 neurons that are so much easier to be able to parse and specifically identify and work with. For electrophysiology, it's totally what you want.
Mm-hmm.
Because you can identify very specific things. So do we think bees are indicative of all sorts of invertebrates? Because, I mean, this is like, hey, invertebrate, you sleep like people.
So bees are a little complicated compared to some other invertebrates because they are able to do very extensive mapping that they can then communicate with one another. And so that's the other thing that they learned through this study was how the brain processes and stores data while it sleeps. And that it's very similar to how our brain works. So the sleep is restorative, but it also provides an opportunity for them to file away the route that they learned that day to the really good, beautiful nectar full
memory
flowers.
Consolidation.
Exactly. Yes. And so they believe that there's a similar process happening in the bees' brains that happens in our brain. That's unclear whether that exists in other invertebrates or not, but I think bees were a really good model species because they knew that this very specific memory pattern had to exist in their brains.
And now I want to know, because we know now that they have the deep sleep that's very much like human brain. Do they have REM sleep? Like it's for humans, rapid eye movement, dogs, cats, etc. Rapid eye movement, but is there an equivalent? And if so, I mean, for an organism that's consolidating memory, the way our brains do it is kind of during that, there's this rapid eye movement portion where the stories, our brain is parsing all the metaphors. It's different. I wonder if bees dream.
I bet they do.
I wonder, yeah. I wonder if their little antenna twitch like a little dog leg.
I bet they do.
And I bet it's really cute.
Yeah.
Thank you for this.
Of course.
You've brought some dolphin stories in the recent past. And I found this particular study really interesting this week, and I thought you'd love to hear it. The question the researchers were trying to ask is, what are dolphins saying?
You don't want to know.
And so, no, nobody really wants to know. But the researchers in this work developed they're using methodologies that have been used previously to record the sounds of calls from individuals, and they do a trapping technique that they're doing anyway on a specific population or specific populations in an area to tag and assess health. And during that process, there's a net collection period where they get a number of dolphins together in a net and then go through them. And what they do is they use like a suction cup on the top of their melon, and record any calls that are made while they are going through this non-invasive process. And so their question in this, not just what are they saying, but this is what they say. They've built a unique library of sounds produced by known individual common bottlenose dolphins. And so what they have are signature whistles, which are the whistles of very specific individuals, like you going, "I'm Blair." I don't know, when you run out into public all the time, "I'm Blair, I'm Blair." That's your signature whistle. And I'm like, "I'm Kiki, I'm Kiki." That's my signature whistle. That dolphins have a signature whistle. And then there are non-signature whistles that all dolphins have. And so they were looking to see the responses after they recorded a whole bunch of different whistles from these dolphins of basically, do the different dolphins have whistles in common, and do the common non-signature whistles seem to correlate with certain behaviors? That would be like a certain, assessment from us humans. And so they have their signature whistles and non-signature whistles. They have resident community of 170 dolphins. And they've been studying these non-signature whistles for a while. And in this particular study, what they discovered is that these bottlenose dolphins, they not only respond to their and other signature whistles, but the way in which they respond to the non-signature whistles is not always the same. And there are certain non-signature whistles that they were saying that they were more like a novelty curious kind of approach, so that these signals, what they call them, like a query, so that this non-signature whistle, it's not just, sometimes it's an alarm function, like, dudes, people vets, what? Alarm. Versus, what? A query, what's going on? So there's the, I heard my name, you called my name kind of responses. But then these non-signature whistles, they call them very creatively non-signature whistle A and non-signature whistle B. Negative is A, that's the alarm, and B is the query. And so I'm really surprised that this is one of the first times that researchers are putting these whistles together in this kind of way, because we've been talking about dolphins as social, vocal, interesting species for a very long time.
I swear we've had studies like this before. We've talked about how dolphins have been assigned names to each other.
Right, so that's the signature whistle. And so
we've talked about vocabulary and syntax amongst different groups before.
That's what I thought. I thought we've done that. Yeah, which is why I wanted to talk about this. I'm like, how is this study saying this is like the first evidence?
Right. There's also I think it's a distinct study that's all about using AI to categorize different dolphin sounds and use it to communicate with them.
Yeah, you brought that story.
That's going on right now.
Yeah. So I found this one really interesting also because they're like capturing the dolphins and using this suction cup hydrophone to record them. And so they're going to get a very limited sample set of calls, right? Like it's going to be, what?
Get this thing off my head.
Get this thing off me. Go away. Like, don't come over here, guys. So the sounds that you're going to get from that, and they do acknowledge, maybe, the next, we got to do a more open, wild animal sampling. But I thought it was really interesting. I think what they were trying to do with the suction cup hydrophones is get like really direct and not underwater transferred recorded vocalizations, but like the actual, all the frequencies and everything involved in the calls that they're doing from their melon, which it is their melon.
Yeah, I'm seeing that there have been previous kind of explanations of what different sounds mean.
Yeah.
This is tough.
And even looking at the spectrograms and the way that they've put this whole study together, which is interesting, so they've done the recording and then they did playback experiments. But they didn't I don't understand how they can say in their title that this is the first evidence of this work. They looked at things like, oh, we played music, we played their songs back at them, and they were ambivalent. And then, that means something. No, they're ignoring you. I thought this was an interesting one from the way it was hyped in the title and how it was being put forward.
I feel like you have to be really careful. Yeah, go ahead.
I would just say that they're not just saying, they're saying that this is the first evidence of possible language-like communication in dolphins. And I am really wondering.
Absolutely not.
How that's true. Mm-hmm.
I feel like whenever I see this is the first time XYZ or scientists have recently discovered the first evidence of XYZ, or anytime it's like the very first or a new thing, I always try to look really carefully to see if there's a specific qualifier on it. So it's like, this is the first time we've studied dolphin language on a Tuesday. Like, you know what I mean? Like, just like some weird variable that makes it a first. But this sounds like they're just claiming that it's a first. And I really feel like we have talked about this several times over many years.
We have. And I've always, we've talked about, dolphins. Oh my gosh, they're talking to each other. What's going on? And of course, they're calling it signature whistles. I'm Blair. I'm Kiki. Then there are the other things.
That's most animal communication. Is hello.
But this is also, beyond that. It's like, does it go further? Do these vocalizations mean more? Is it not just alert or query?
Definitely. If they play,
where does it go?
They play, they have culture. They're like, I want to use this sponge. Well, I want to use this sponge. They're communicating. There's very intense communication happening. There's also, beyond all of the things that you mentioned, there's the clicks and the squeaks and all the other things that people who work directly with dolphins know what those things mean, which means it is language.
All of it inclusively together is part and parcel, right? So anyway, final point on this. If you see it in the news, this is a bioarchive study, has not been peer reviewed. So hopefully it will be peer reviewed and there will be, maybe some adjustments before it's fully published. But yeah, there's a whole lot of issues with that study. But have we talked about programmed cell death a lot? I love programmed cell death, apoptosis. When things go to a certain state in your cells, calcium channels open and then it's like, oh no, the calcium meter's too high. And so the cell goes, well, that's it for me. And a whole process of mechanisms take place. And then the cell dies. And this is good.
It reminds me of, just because it's easier for me to picture big things and then scale them down, how octopuses go into senescence after they reproduce. It's basically programmed cell death system-wide. Right.
Yeah. And so programmed cell death on little scales, or even sometimes on organ level scales, sometimes it works, it's right. And you need to have it as part of development, not just death. It's part of life. And this programmed cell death is an important process for keeping an organism healthy. However, sometimes it's involved in things like Alzheimer's disease or Parkinson's or, other neurodegenerative diseases. And so researchers at Melbourne, at Walter and Eliza Hall Institute in Melbourne, Australia, just published in Science Advances their work trying to stop it. Because, hey, why stop the cause when you can stop the process? So if you see lots of cell death, the cell death potentially is part of in neurodegeneration, your brain, parts of your brain dying off. And when your neurons die off, then you lose the things that those neurons did. And so if you could block it, maybe that would be good. I don't know. I am actually I question this methodology because I don't know that it would be better to block cell death if the processes that are leading to cell death would negatively impact those neurons in other ways that would be bad. Like, is cell death is this is an interesting question. But anyway, the researchers have found a target, a protein called BAX, that there's a killer protein called BAX, and it kills cells by damaging mitochondria. But it's part of the apoptosis programmed cell death process. And so these researchers were like, ooh, let's look at a whole bunch of molecules, chemicals, etc. And they found one that is specific to hitting BAX. And when it hits BAX, it makes BAX stop. And so BAX can't turn off mitochondria. And so I think this is really interesting in the large scale conversation that we've had over the last few years about the importance of mitochondria and mitochondrial health to aging, right? And so there's this whole systemic question, right? Where it's like, okay, there's something wrong with maybe the garbage disposal system in the brain, maybe something wrong with mitochondria. I don't know. But they are broken. And so because they're broken, apoptosis starts and BAX comes in and goes, okay, you're done now. And the cell needs to die. But if you just stop BAX from working, the broken stuff keeps going.
Mm-hmm. Yeah, this is what I'm concerned about. You're like,
you don't have neurodegeneration. You just have you just have less functional neurons.
You're treating the symptom and not the cause, basically, right?
Mm-hmm.
You're putting a Band-Aid on a cut, but your blood won't clot. You know what I mean? Like, you're like, oh, I'm fixing the problem. No, you're fixing the symptom, not the problem. Similarly, it feels too broad because some cells need to go. There are conditions where too many cells are going, but if you stop all of it, you're going to have huge problems.
Yeah. Yep. Yeah. So
I keep vacuuming up Legos, so I stopped vacuuming the house entirely instead of picking up the Legos.
Instead of having my child pick them up.
Yeah.
Yeah. So, I mean, what I'm wondering, I think it's good to understand potential targets and places to act, but I'm wondering if they get it past this just general understanding phase, is this a worthy target for therapeutics? And would it work with other, drugs that people are taking for their, symptoms?
And I guess ultimately, if you're going to die from Parkinson's or Alzheimer's, you could do with some junk cells if you're not going to die, right? Like it's a trade-off in this case.
And that's the question. Like, we don't know. All we know right now is they found this, like, ooh, we could block apoptosis. We could stop this molecule from destroying mitochondria that are probably, if they don't die, going to impact the cell in other negative ways, because like the reason you have apoptosis is so that the cell doesn't go out of control. Like, it's a control factor. But this is one of those things where it's like, oh, let's just put like rabbits in Australia and see what happens.
So if you stop cell death, do you stop making new cells?
No.
So that's bad too, right?
Yeah. I mean, as you age, you have an increase in cell death. Not necessarily. It depends on what stage of development. But you also have a slowing of cell birth as well as you're aging, but not stopping. It doesn't stop. And so, yeah. All of this is like when you have the disease states. I mean, the ideal would be to figure out exactly what's happening with the disease state and try and figure out how to target the cause. Is it the mitochondria? Is it, the energy function? Is it the garbage? Is it your glial cells not working? Or, what else is going on? As opposed to, hey, let's just let the broken system keep working and see if that works. So you can limp along. But yeah, nobody knows anything yet. This is simply a study that has figured out this mechanism with the idea that they're selling behind it of like, maybe we could use this to stop apoptosis. And I'm sitting here going, please don't. Please don't. Oh my gosh, why would you do that? But I'm not in charge.
Well, rat study's coming next. Let's see.
We'll see
what happens.
What about teeth?
Yeah.
You like them?
I love having teeth.
You know that they probably came from armored exoskeletons of ancient
Yes, I
fish?
did know that.
Yeah. That's really cool. All right, great. We're done.
No, actually, I think that I thought what I was taught in evolutionary biology was that they were most likely scales.
Yes. So skates, rays, they have, and sharks, they have these tooth-like
dermal denticles.
Denticles. I love that word. Yes. So this is something that we know. However, researchers just publishing in Nature, a researcher at the University of Chicago in Neil Shubin's lab was like, hey, I want to see where I'm looking for actually what is the earliest vertebrate in the fossil record. And so this researcher, Yara Harity, took a whole bunch of fossil specimens that she could identify as vertebrates because they had these bumps, these denticles on the outside of them. They're called odontodes. There's this sign is the presence of dentin in these bumps in their external armor, odontodes. She got a bunch of specimens. She took them to Argonne National Laboratory, advanced photon source, and stayed up all night. She says, it was a night at the particle accelerator. That was fun. And they did high resolution CT scans of all of these fossils. And what she found was not the earliest evidence of a vertebrate in the fossil record. What she found is evidence of where the very first tooth-like structure in vertebrate tissues came from. So they were looking at all of these, and they found one sample from the Cambrian fossil called Anatolepsis. And it looked like it had the odontodes and everything. But then they looked at it, and they're like, oh my God. And so they're looking at things. They found some that had the dentin and others that didn't. There's one fossil that they thought was a vertebrate, and everybody thinks it's a vertebrate, but then they found it didn't have dentin in it, and they're like, oh my gosh, it's an invertebrate. So they completely moved a fossil to another part of the taxa. But anyway, these dentin-like tubules. That's where it all came from. And teeth, they say, this shows us that teeth can also be sensory, even when they're not in the mouth, is what Harity says. So there's sensitive armor in these fish. There's sensitive armor in these arthropods, and it explains the confusion with these early Cambrian animals. People thought it was the earliest vertebrate, but actually it was an arthropod.
Yes.
So
very
sometimes Sometimes we all need to spend the night at Argonne National Laboratory doing CT scans of ancient fossils to look for evidence of things that we did, asking one question, but instead follow the evidence and answer another. So it is, yeah. Pepsi Cola in the chat is saying lucky find, and that is, I think, one of the things about this study that I think is great is the researcher Harity didn't focus on what they wanted out of the fossils; they followed what they got, and so, yeah. So we all get these denticles, odontodes, and denticles. I love these words too. Right? They're great. But the teeth in your mouth, as sensitive as they are, at one point, maybe there were similar cells and instructions on the outside of your ancestor's body saying, we're going to have sensitive teeth on the outside.
So just imagine next time you're running your tongue across your teeth, what if your teeth were made of skin?
But it's not skin.
It's the equivalent because it's on the outside of the body, right? So
yeah, exactly.
It's sensitive like our skin is.
No, I went the creepy direction, which is what if we were covered in teeth?
I think that looked cool. They just look like white scales, kind of like a decoupage. Is that the right word?
Do you like my teeth coat? What do you think? What do you think of my teeth today?
Yeah, decoupage. I think that was right.
But what if we had our whole body's covered in teeth, and then like everyone's like, "Oh, I gotta go to the store and get like those bleach strips to make sure I have wet teeth."
Oh man!
Never a dull moment. Have we done it, Blair?
Oh yeah. Can you imagine getting a cavity on your skin? All right. Yes. Now we're done.
A lot of toothpaste. You don't need soap in the shower. You just need toothpaste.
Anyway.
Oh,
yeah, no, we've done it. We're all good.
I could make jokes about teeth coats all day. Everybody. Blair, thank you for tonight.
Of course. Thank you. What a fun night.
Yeah, it was good. Everybody, thank you for joining us for this episode of TWIS. We're so glad that you're here. I hope you had a wonderful night. And now I want to thank especially certain people. Those of you in the chat room, oh my gosh, thank you for being here and chatting and being just wonderful, wonderful people. Thank you for being real and compassionate and respectful. Really love that. Gord, Arnlore, others who help with making sure everyone is being respectful and it's a good place to be. Thank you. Thank you for being here for so long too. Identity 4, thank you for recording the show. Fada, thank you for help with show notes and with social media. I really appreciate it. And Rachel, thank you for editing the show. And of course, thank you to our Patreon sponsors. It is time for me to say thank you to Robert Norlin, Robert W. Farley, Lauren Gifford, Dana Lewis, Eden Mundell, Alan Viola, Aaron Nathma, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, George Corris, Pierre Velazarb, John Ratnaswamy, Chris Wozniak, Bigard Shefstad, Donovan Styles, a.k.a. Don Stylo. Oops, hit a button there. What's happening? I don't know. Allie Coffin, Shubrew, Sarah Forfar, Old Boomer 847, PIG, Stephen Oliveron, Daryl Myshak, Andrew Swazin, Fred S104, Sky, Luke, Paul Ronovich, Kevin Reardon, Noodles Jack, Ryan Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Cassenblow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Clemmy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Phillips, Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, Lon Makes, EO, Adam Mishkon, Kevin Parachan, Bob Calder, Marjorie, Paul D. Disney, Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon. And if anyone else would like to support us on Patreon and have their name read after the show, head over to twist.org and click on that Patreon link on next week's show.
We will be back on Wednesday at 8 p.m. Pacific Time, broadcasting live from our Twitch, YouTube, and Facebook channels. Hey, do you want to listen to us as a podcast? Perhaps while you study bee brains? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well. Should I keep going? Sure.
Sure.
Right? Yeah. For more information on anything you've heard here today, show notes and links to stories will be available on our website. That's at www.twist.org. I see a couple of you in the chat room asking for links to stories. That's how to get them. And you can also sign up for a newsletter when that pops up. It'll arrive somewhere, somehow. It'll be a surprise. We love your feedback. If there's a topic you would like us to cover or address, or a suggestion for an interview, please let us know on one of our social media accounts, or better yet, send us an email. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into a red fluorescent spider web. We look forward to discussing science with you again next week. And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth.
And what's up, Blair? And it's the after show. It's a tired time. Blair.
Yes. Sorry.
No. Yawn. And bring the realness in. I just want all of us to know that we're going to talk about real things. We're going to talk about the politics sometimes and how it interacts with science and the fact that we're all involved in it. And all of it, any system that people have is made by people. And we are part of constructing that system. And don't ever forget that you're a part of it.
Yeah.
And remember that hope is rebellious.
Part of the reason things are moving so fast right now is because I think a lot of people know that come midterms, when everyone's really upset about everything that's happening, suddenly the power to do pretty much anything is likely to be taken away. But we need to make sure that that actually occurs. So that's what's really important, is that the people who want to kind of do a slash and burn of everything that this country's built that is productive, that they are blocked at every opportunity as soon as we have the opportunity to put some people in power to do that. So, yeah.
And science has been a big part of what's being impacted. The EPA, all sorts of things that are holding their efforts back.
I think that's what was so depressing about the Endangered Species Act release, was that this edit was released by Fish and Wildlife, and I think it was NOAA. So it's because
it comes with
there are people being appointed,
title,
and then there's still people below them who got into that line of work because they're passionate about it, but they're stuck now with a person who's been appointed to the top level there, who's just going to do whatever they're told. And do you stick it out and try to fight the good fight from within, or do you bail? And it depends on what your goals are, and it depends on what your personal mental capacity is to do that. And it depends what your short-term and your long-term goals are, right? So it's, I've heard stories about people who've worked for 20 years to get their job at the National Park Service, and then they finally got it, and then they
Bye-bye.
were fired.
Gone.
Yeah.
Mm-hmm.
I've also heard of other people who spent their entire career getting to work at a government research center that bye-bye, or fundamentally changed, had to step away voluntarily, right? So, yeah, sometimes you can stay and try to make change from within. Sometimes you cannot. That's a very personal choice.
It is. And it is whatever is right for you. And it is, both are okay. It's just, and so that's what I hope, is that there isn't too much splintering because of judgment, right?
You know what I think about all the time?
There's a need for people who stay and
Yes.
stay within, and little things are just part of it every day. And there's a place for the leaving and the big statements and like saying, I can't do anything there anymore, and I'm not going to be a part of that, right? It's both.
Yeah. There's it's kind of older now, but there was something called The Story of Stuff. Do you remember this?
Yeah, I do.
So they made a second video. It was called The Story of Change. It was a whole package related to that. And I think about it all the time, because they said that everybody has a different role to play in a change movement. And so you don't have to be the one on the street picketing and screaming. You might be the person cooking meals for the picketers. You might be the person designing the signs that they hold. You might be the person writing letters to your congressperson. There's all these different roles that you can hold as a change maker, and they don't all look the same.
Yeah.
The other thing I think about all the time, and I know I've mentioned on the show before, is there's this TED Talk that I have bookmarked on my work computer. I might be able to find it, but it's How to Start a Movement. And it's all about the idea that, of course, in order to start a movement, you need that first person, right? But actually, the second person is more important than the first person, because the first person is a weirdo doing their own thing on their own. The second person creates buy-in. The second person makes joining in acceptable. So being the second person actually requires more bravery and has more influence on all of the people that follow them. And so I think about that one all the time as well. You don't have to be the person to come up with the activity and to start doing it all by yourself. You can look around and find things in progress and join them. And so everybody has their own role to play. Maybe you are the person, you are the visionary who's going to start a movement as the initial person, but maybe you're number 2 or maybe you're number 10. And we need everybody.
And maybe you're someone who your role is you just support people, and that is so important. Like, yep. I saw your unhappiness tonight. I know you're also tired, so we're gonna close it up. But I want to say, take care of yourself, because you can't let that take you down. So take a break if you have to. Do what you need to. Do what you need to. I love that you were here tonight, because we got to talk about spiders and shrinking fish and like some other fun stuff mixed in with the serious. And that's what I love about this show on a weekly basis, that it kind of often pulls me out of my little funks, because it reminds me that I'm not alone.
That's right. We are not alone. We're all angry. Actually, I was really mad when I was researching these stories earlier today. And then I started reading the comments, the public comments.
other people
And I was
saying
getting
things we're
less and
thinking.
less mad because I was like, okay, like this is truly just a room full of people trying to get rich that have no regard for public consensus. But that has happened time and time again with this administration. They are going against public consensus on all sorts of things.
Yep.
So
And I
it's
wonder
going to make
if
them money.
it is. And I wonder if it's the thing because they stopped paying attention and they're just trying to, you're going to listen to my double speak, and that's fine. That's all that is. Great, great. But when you take away people's, VA care, Medicare, Social Security, retirement plans, like, insurance, like all these things that help, especially at the lower ends of the economic scale.
It's really hard. I very consistently have to remind myself, like you were saying, to like breathe, take a break, and focus on things that I have control over, because it is really scary. Like all of the things that you mentioned, people are going to die. People are going to die. And the callousness with which those decisions are made is really disturbing.
That's it, exactly. Yeah. It's not the same.
And now
we can't be in that mindset because we're not. That's a different.
Right. I can't imagine that. And now sweeping decisions that will destroy entire habitats and entire species. We could lose species forever from this decision. And in the 70s, 50 years ago, like people were still like, is smoking bad? I'm not sure. Like 50 years ago. But we decided, oh crap, the bald eagle's almost gone. Let's do something about it. That happened then. And now we're like, screw it. Like, that's irreversible. I think that's the thing that's really scary, is it is irreversible. Like
let's go back to the 50s, Blair. Come on. Who cares about the 70s? It's the 50s.
Oh yeah, because neither of us are supposed to have a job or on credit card or wear pants. So just or access to birth control or any
of the things. Yeah, it's the 50s, Blair. It's great. Life is great. It's the best it's ever been. It's the best.
The good
Don't we want
days.
it the best again?
Yeah.
When people smoked without care, giving other people cancer who were just in the room all day. When people drank 3 martini lunches all the time.
And don't forget when segregation was still a thing.
When segregation was a huge thing. When people didn't have like complete voting rights. When that life was great. Let's do it again. I'm gonna say, let's not. Can we not? Okay.
Yeah, let's not. I just keep thinking about
better. We know so much more.
The meme of I just want to live in precedented times. But
instead of unprecedented.
Yes. Yeah. This will be a chapter in a history book that will be taught forever and it'll have some weird name like the Great Constitutional Tumult or something like that, right? Like somebody will give it some name, like the Teapot Dome scandal, where everyone will have to learn about it in school. I still remember those words. Do not remember what they meant at all, by the way. Do you remember what the Teapot Dome scandal was?
No, I don't think I learned that in high school.
Really?
That's not something my high school taught me.
Okay, let me look.
Come on, Blair. I grew up out in the country.
It was a bribery and corruption scandal that rocked the Harding administration in the 1920s. Secretary of the Interior Albert Fall, who illegally leased U.S. government-owned oil reserves at Teapot Dome, Wyoming, and other locations to private oil companies, including those of oil magnates Harry Sinclair and Edward Doheny, in exchange for substantial bribes. Fall was convicted of bribery and became the first cabinet member to go to prison.
What?
There you go. Teapot Dome scandal.
It happened before. It could
Yeah,
happen
yeah.
again.
Yeah, yeah,
Sending
yeah, yeah, yeah.
cabinet members to prison. What?
Yep, yep. So, anywho,
Huh?
this whole period of time is going to have some funny name. It will, it'll be talked about, and then the following chapter will be all about how it all had to get systemically fixed.
Exactly. And how it
So
changed
I just want to get to
our
that
country's
chapter.
culture
I want to get to the
forever.
fixing chapter. It's fine. We'll figure out. We'll deal with the fallout. Just can we move on now? Can we stop messing things up worse and at least just flatline for a second so we could start to make things better again? And really, that's not going to happen until 2026 at the very, very earliest. Well, 2027, I guess, because the election will be in 26.
And then when people come into office.
Yeah, we'll be early 27. So it's, just a year and a half.
It's just a year and a half. And in the meantime, take breaks, love the people you love, appreciate the moments you have gratitude for all the things you have and the place you are in your life. And when you have the chance, have compassionate, respectful conversations with people.
Oop, I just lost your audio again.
Doesn't like me. Am I here?
Now it's back.
Okay.
What in the heck?
Yeah, have compassionate, respectful
There
conversations
you
with
go.
people
Yes.
about how science is a part of their lives and it helps them live better lives. That funding from the government and not necessarily private sources allows a more consistent, reliable source of funding to make the advances that are going to get rid of cancer, that are going to help us live better, get rid of diabetes, like all the things that you wish would help us live happier, better lives. Government funding helps get us there. It really, like basic science, DOE, NSF, NIH, basic science to applied science, the way that it moves through, if we have government funding, it is a reliable source. And that's the problem with what's happened, is that all of a sudden, all this work, all this stuff became unreliable. The system became unreliable. We don't even know. A bunch of philanthropies are like, oh yeah, okay, we'll make up for it. It's not all going to make up for it. The system has been disrupted. So it's now uncertain times. So when you have a conversation with people and then they're like, oh yeah, whatever, we don't need to spend that much money in the government, who cares? Then you're like, oh yeah, didn't your certain loved one have such and such a disease, and weren't they cured by this treatment, whatever, and wasn't that great? Don't they know how to, like this, how, oh, that was funded by the NSF, by the NIH. Oh, wait, oh, you didn't die of salmonella or E. coli in your salad? That's because of the FDA, right? It's part of regulation and funding the infrastructure and make it possible. Oh, we're not all going to die from bird cow flu? That's because we kept funding.
How about an
The whole
entire
system.
school didn't end up with scurvy because they decided that ketchup was the only vegetable they were going to provide?
Right? I know. Exactly. When people make decisions on whims and opinions and impact everybody else, that's not the way that we want things decided. But we all need to talk about it, but not talk about it aggressively.
Set a time limit is my suggestion. I have a time limit for my news every day.
Time limit, yeah, for looking
I
at
can't
news,
do it all day.
But even a conversation, it's like, oh, okay, a little time. Oh, okay, I'm getting nowhere. I'm done now. Let's change the topic. Don't feel like you need to change a mind. You do not need to change your mind. All you need to do is start a conversation. And maybe the conversation will continue.
Plant the seed.
We're all gardeners of knowledge.
Yeah, we are.
Yeah. Blair,
Yes.
you're a good gardener. Say goodnight.
Good night, Blair. Say good night, Kiki.
Good night, Kiki. Good night, everyone.
Good night, everybody.
Thank you for joining us for another episode. I hope you all get a good rest this week. Stay safe, stay healthy, stay curious. Never stop asking questions. And stay lucky, because that's great. Good night. Thank you.